All right, so now what we're going to do is talk about some microbial genetics. And so this chapter, chapter 11, is actually going to be quite a bit of review from just regular biology classes and other prerequisites. So what we'll do is we'll go through and we'll talk about the different pieces that are specific to microorganisms, but we'll kind of go pretty quickly through some of the stuff that's universal that you should have already covered in other classes. So it'll make this chapter a little less daunting, or a little less large, I guess, because it should be a lot of information you already know. So we're going to talk about the functions of genetic material, and we'll take a look at DNA replication and transcription and translation, which all of those things which you should already know, and you should probably already know some about mutations. So point 2, point 3, point 4, and point 5 should be a lot of review with some bacterial or other microorganisms peppered in. And then we'll talk about a lot more specific to microorganisms here on 11.6 and 11.7. So let's start out here. The introduction here is talking generally about how, here's a quote, "What is true in E. coli is true in the elephant," as it says here, suggesting that the biochemistry of life is maintained throughout evolution and is shared in all forms of known life. So we've learned a lot, and we know that there are lots of common things here. That's why a lot of this will be repetitive, things that you've already learned. However, there are significant differences among the three domains of life. So that's why we're going to talk about the differences between eukarya, archaea, and bacteria, but we'll also see a lot of similarities. We will also touch on viruses, because they are cellular parasites, meaning that they aren't living things on their own. They have to be within a host, but they do show dramatic variation in their genetic material and their gene expression processes. So we will take a look at these different types of things. So 11.1 is just a general overview, the functions of genetic material, which should be a bit of a review. So DNA serves two essential functions. So first, DNA is the genetic material responsible for inheritance and is passed from parent to offspring for all life on Earth. So number one, DNA is something that we pass down to create more living things. It has to be replicated very accurately, hopefully with minimal errors when it's passed down. And it's organized into smaller discrete units called genes that are arranged on chromosomes, and then also in plasmids. As we saw in the previous chapter, we have plasmids, which are a type of extra chromosomal DNA that we see in bacteria, for example, not so much in eukaryotic organisms like humans. Then our second function of DNA is to direct and regulate the construction of proteins necessary for cell growth and reproduction in a particular cellular environment. So in that vein there, we're going to talk about the second function in this chapter. So a gene, remember, is composed of DNA that is read or transcribed to produce an RNA molecule during the process of transcription. So transcription, again, is transcribing, meaning writing down, going from DNA, writing it down to RNA. So our major type of RNA molecule, again, is called messenger RNA or mRNA. And then this provides the information for the ribosome to catalyze protein synthesis. And when we take our mRNA and then we go through the ribosome, making our protein, that's called translation. So we're translating what is found on the messenger RNA and translating it into an actual three-dimensional physical protein. So the process of transcription and translation are called gene expression. So going from DNA all the way to our protein is called gene expression, so through transcription and translation. So gene expression is the synthesis of a specific protein with a sequence of amino acids that is encoded in a gene. So going from gene to protein is gene expression. And this is also the central dogma of biology. So the flow of genetic information from DNA to RNA to protein is the central dogma of molecular biology specifically, and talks about Beadle and Tatum, which we haven't gone into detail with. So each of the processes of replication, transcription, and translation includes these stages, and we'll be talking about them in more detail. So first, we have initiation, then we have elongation, also known as polymerization, and then termination. And we're going to take a look at these 3 stages, and we'll see some differences between eukaryotic organisms and prokaryotic organisms. So down here we just have kind of an illustration of that. So this is our central dogma, this is our gene expression. So we're going from DNA, which would have the gene in it. We're going through transcription, so we're transcribing it into RNA, and then we translate that RNA into our protein. So our cell's genotype is the full collections of genes it contains. So our genotype are the genes, and then we have our phenotype, which is our observable characteristics that we get from those genes. So genotype, pretty easy to remember, is our actual genes that stay the same within our body. And then our phenotype, I think of as physical, what it physically looks like, rather than the genotype. So the phenotype is the product of the array of proteins being produced by the cell at a given time. So this is, of course, influenced by the cell's genotype, as well as, though, interactions within the cell's environment. So within our own body, we see this very slightly, but then in bacteria we may see this in greater amounts. So we have our genotype, which are actual genes, the letters of our DNA. And then what we'll see here is that we have genes that can code for different things. And if we find ourselves in environmental condition A, for example, then we might see phenotype A. Versus if we find ourselves in environmental condition B, we might see phenotype B. And again, this is seen most often in prokaryotic organisms where they can shift and change based on the environment. They're much smaller. We do see that within ourselves, but not to the degree that we see it or as easily as it is seen in bacteria, for example. So again, genes code for proteins that have functions in the cell. A specific protein encoded by an individual gene often results in a distinct phenotype for the cell compared with the phenotype without that protein. So it's common to refer to the genotype of an individual gene and its phenotype. So it's important to understand that we have the distance or the differences between genotype and phenotype. So our genotype remains constant, but we don't have all of these proteins being made all of the time. So they're not all going through this protein production simultaneously. We carefully regulate the expression of these genes, and we only make the specific proteins when those specific proteins are needed. So that's why we would see phenotype A or phenotype B. And I think there's an image of Serratia marquescens, maybe it's further on here, where it shows that Serratia marquescens actually, when you grow it in a lower temperature, about 25 or 27 degrees C, then you see that it is a red growth on the agar plate. If you grow it at higher temperatures, like 37 degrees C, then it's a clear type, or foggy type of growth on the agar plate. So you really see a difference, even just in the color of the organism at the temperature that it's grown. And that would be an example of having the specific genotype, which doesn't change, but environmental condition A might be our lower temperature, and then our phenotype A would be red coloring. So this would be like, say, 25 degrees C for Serratia marquescens, and then its phenotype would be red. And then environmental condition B might be 37 degrees C, and then the phenotype would be creamy or cloudy. And so then we would have the same genotype, but then different phenotypes in that case.